I'm Dr. Lauren Beretich, board certified genetic counselor, PhD scientist, and member of ASGCT's Patient Outreach Committee. Did you know genetic testing results can be a key piece of information used to determine whether a patient qualifies for a clinical trial? I want to spend the next few minutes talking about just that, genetic testing and how it impacts clinical trial eligibility. As you may know, genetic testing is a broad term referring to different ways to analyze our genetic information, or DNA, to better understand if it is typical or atypical. Imagine your DNA as a cookbook that contains recipes or instructions for making every part of your body. Genetic testing looks at this cookbook to see if there are any typos or mistakes in the recipes that could lead to health problems. Genetic testing continues to be incorporated into various healthcare specialties and is commonly used for the purposes of family planning, predicting one's risk of a particular condition, and making a diagnosis. While barriers still exist, genetic testing has become more accessible and convenient. Many genetic tests can be conducted on blood, buccal, or saliva samples in a doctor's office or even at home. It's important to remember that while genetic testing can help healthcare professionals make a diagnosis, results can also be uninformative or indeterminate. Because of the complexities, it's important to speak with a genetic counselor or other qualified healthcare professional regarding your genetic testing results. If needed, you can locate a genetic counselor online at findageneticcounselor.com. In relation to clinical trials in gene therapy, genetic testing is often important to determine if a patient meets relevant inclusion or exclusion criteria. For many genetic conditions, genetic testing is necessary to establish a diagnosis or confirm a suspected diagnosis based on an individual's clinical features. For example, in a gene therapy clinical trial for an inherited retinal disease, Leber congenital amaurosis, a confirmed genetic diagnosis based on RPE65 gene variants was a key inclusion criteria. In addition, genetic testing provides variant-specific information. A therapy or potential therapy may be best suited for individuals with particular genetic variants, or may be contraindicated for others based on the therapy's mechanism of action or potential adverse reactions. For example, in a clinical trial for Duchenne Muscular Dystrophy, or DMD, the gene therapy was designated for individuals with specific variants in the dystrophin gene, particularly those between exons 18 and 79, excluded those fully contained within exon 45. Individuals with variants outside this range were less likely to have a safe and effective response to the treatment. For clinical trial teams and other managing healthcare professionals, genetic testing results may be helpful for prognostication or understanding how one's condition is anticipated to progress over time. For example, in trials for spinal muscular atrophy, or SMA, genetic testing can provide information on the type of SMA and its likely progression, helping researchers of the healthcare community interpret clinical trial results and evaluate the effectiveness of new therapies. Understanding a patient's prognosis without treatment aids in assessing how well new therapies work compared to the natural progression of the disease, especially in the absence of placebo or control groups. As we discussed, genetic testing can be critical to determine one's eligibility for a clinical trial, especially cutting-edge genetic medicines. Offering genetic testing has vast diagnostic and clinical utility, including identifying which clinical trials may be most beneficial. Thank you for watching, and for more information on gene and cell therapy, watch more of our community quick takes.
I'm Kevin Flanigan, the director of the Center for Gene Therapy at Nationwide Children's Hospital in Columbus, Ohio, and a neuromuscular physician and trialist there as well. Question is, how does the location of the mutation in the DMD gene impact whether a patient can participate in a trial of gene replacement therapy for Duchenne muscular dystrophy? It's important to remember that many of the mutations in the dystrophin gene are mutations that delete a portion of the gene. so that there's no mechanism by which a boy affected by Duchenne muscular dystrophy with that type of mutation could ever have expressed that portion of the gene, that portion of the protein encoded by the missing portion, could never have expressed it in their body. So if that is now re-expressed, it's re-expressed by putting in a transgene that expresses that missing portion, there's the risk of an immune response to it. This idea, sometimes you call it a novel epitope. An epitope is just the portion of a gene that induces an immune response. So if the body is seeing a novel epitope, then it has the risk of having an immune response to it. One way of thinking about this, a colleague of mine frequently refers to it as a single molecule transplant in a way. We put in this single molecule that contains now a new version of dystrophin. If the body's never seen the portions of that protein that are now expressed, there's this risk of an immune response. So the importance of this idea has actually been brought about by a very nice effort among multiple companies doing gene therapy trials in the Duchenne space to share some of their data. And in fact, that analysis of that data resulted in a publication about a year ago, in mid-2023, in the New England Journal of Medicine. This is a figure from that paper. And although it looks a little complicated, it explains these observations that demonstrated the point. So this is a cartoon of the structure of the dystrophin protein. The structure consists of what we call the amino-terminal binding domain, the carboxy-terminal domain, and this long section of modular spectrin-like repeats demarcated by various hinge regions of the protein. And under this is mapped kind of the structure of the 79 exons of the Duchenne gene, both what they are sort of idealized there, marked by all of these exons, but more importantly, how each of these constructs of different gene, microdystrophin genes, transgenes are mapped to it. Each of these they all share some portion of the N-terminus, the front end of the protein. They share some portion of the C-terminus, but there are gaps that these missing portions are regions of the gene that are not included in the transgene. What was observed comparing among patients from multiple studies, at this point, there were 5 patients who had been well-defined who had inflammatory responses, either in cardiac muscle or in skeletal muscle, marked by skeletal myositis or myocarditis. That inflammatory response was a kind of T-cell immediate immunity, the kind of immune cells that may attack cells within the body that are expressing new antigens. One thing that all of the patients who had these responses shared in common was the presence of deletions that extended into exons 8 and variations of these in each of these cases, but all of the transgenes shared a region of expressing a neoantigen, a new epitope that covered this region as well. So this led to the idea that exons 8 through 11 might be particularly immunogenic. And further studies have sort of carried this forward as well, suggesting that this region, encompassing the first hinge region of the protein, for whatever reasons, is particularly immunogenic. So for this reason, several of the trials, the ongoing trials for microdystrophin therapies, exclude patients who have mutations in this particular region of the gene, but don't express, who have deletions in this particular region of the gene. Some of them are limiting as well for patients who have mutations anywhere from 1 through 17 as well. Another approach that might be possible in gene therapy to avoid these sort of immune responses is to provide anti or immune modulating drugs prior to delivering the gene therapy in a way to diminish the possibility of having this kind of immune response. That's also an option for some trials. So some future trials may not have these exclusions. I'll note, though, that the recently approved gene therapy, microdystrophin gene therapy, does issue warnings about using it in this region of the gene, this N-terminal region of the gene, where mutations are present in the N-terminal region of the gene, I should say.
In regards to the question of how genetic testing can impact the immune modulation that we use in clinical trial participants, this is really an important consideration, both for just how we think about eligibility for clinical trials or for even dosing with commercially available products, and for thinking about the potential that the immune system may play, and how that might impact, for example, types of immune modulation that we might need to use. So as a first step, genetic testing is very helpful and necessary to confirm a diagnosis and ensure that we, as the clinical team, can provide an accurate diagnosis, properly review the various care considerations and needs for a given individual, and have a meaningful discussion around the different possible therapy options, which in some conditions may include gene delivery or gene transfer therapy. When using adeno-associated virus for the delivery of a gene, a key aim is for the production of a new transgene protein or product that was otherwise either not functioning or missing in a participant or patient. And as an example with giant axonal neuropathy, as is also an issue for many other genetic conditions, there is a potential concern about whether the new transgene product will be considered foreign by an individual's immune system. Having genetic testing can allow us to stratify individuals based on their genetic changes in the GAN gene, for example, to estimate whether we expect that they may produce the gigaxonin protein or not at baseline. In the scenario where some patients or participants have what we call biallelic null variants, such as deletions, out-of-frame changes, or other mutations that may lead to an early or premature stop on both alleles, we worry that an individual may be at greater risk for developing an anti-transgene response following gene transfer than those individuals, for example, who might have missense variants and who may express some protein, albeit still dysfunctional, thus leading to disease. There is always the possibility that even additional patients could be at risk for a response against the new transgene protein, even regardless of the genotype. And so it is important that we think about monitoring for this in all participants or patients who are receiving gene replacement therapy. There can be tests, for example, that are performed in the scope of gene therapy clinical trials to check for anti-transgene responses. This could include antibody testing or perhaps looking for specific T-cell responses. In GAN, this monitoring was done by evaluating T-cell responses by a test called an ELISpot analysis that was performed from a blood sample. So beyond trying to understand who could be at risk for an immune response to the transgene protein, a next step is trying to understand what medications or what factors can we use to mitigate or hopefully decrease that risk. Again, the first step is going to be to stratify whether someone needs an additional immune regimen or not, based on review of their genetic variants. In some diseases, there may be the additional ability to evaluate for levels of an expressed protein or an enzyme that may also further help with that. But in cases where that is not a scenario or an option, we try to model this based on review of the genetic variants based on their genetic testing that had been performed. Again, we do not want participants who have received gene replacement therapy to develop an anti-transgene response. You could sort of envision this response as a body's rejection of the new protein that their body is now producing. And this can then potentially decrease the effect or benefit of the gene transfer altogether, and can also lead to safety concerns related to this heightened immune response. Thus, to prevent this response, in the GAN gene transfer study, we implemented an additional immune suppression protocol for those individuals who have what we call biallelic null mutations, or also termed predictive Crim negative. In addition to using corticosteroids that were used in all participants, those participants who were deemed at a higher risk for an anti-transgene response also received medications that would help target the T cell population or T cell responses. And this included medications like rapamycin and tacrolimus. These are medications that have also been used to treat other conditions, including for organ transplantation, in order to reduce the risk of rejection. Interestingly, rapamycin may also have some additional benefit in working on part of the immune system that promotes something called tolerance. That is a process by which your body may select a protein as self or acceptable rather than foreign, and thus hopefully reducing the risk of mounting an immune response against that protein. Overall, another question is when to receive the immune modulation. When using immune modulation strategies, participants receive immune therapies at different time points. For example, in the GAN clinical trial, we had some of the immune therapies starting shortly before gene transfer, and then continued immune modulation after gene therapy for extended periods of time, in some cases months, or in the case of rapamycin, potentially even longer, depending again on the genetics or genotype. In the field of gene transfer, I'm not sure if we yet know what the ideal length of time for T cell targeted immune modulation may be. There can be challenges as to knowing, for example, if an anti-transgene could occur later, how quickly it may develop if it occurs, or how it will manifest. Thus, it remains imperative for individuals who are receiving gene therapy, whether it's in trials or even in clinics for commercially approved therapies, to have very close follow-up, not only in the short term after gene transfer, but also for longer periods of time. This will allow researchers and clinicians to be able to track a given patient or participant's health, their function, and other lab parameters to hopefully be able to identify a response early should that occur. I think we're at a truly exciting period of time for the field of genetic therapeutics, but they continue to require caution, close monitoring, and vigilance. In addition, our ability to share experiences between diseases may allow us as a field to understand these responses should they occur. For example, a lesson learned in a gene therapy clinical trial for a genetic eye disease or another blood disorder, perhaps like hemophilia, could allow us to learn about effective immune regimens, treatments, labs, or monitoring in patients with neuromuscular or neurogenetic diseases as well.
Hi, I'm Kim Goodspeed. I'm a child neurologist by training and neurodevelopmental disability specialist. I'm currently working with the company Ultragenyx on the Angelman program, where we have an antisense oligonucleotide that we're working on for Angelman Syndrome. But I also serve as adjunct faculty at UT Southwestern in Dallas, Texas. So Angelman syndrome is an imprinting disorder. It's a really interesting genetic condition because in most of the body, UBE3A, the gene that causes Angelman syndrome, is expressed from both copies, like any normal gene in your body. But in brain tissue and in spinal tissue, so in neurons, only the maternal copy, the mom's copy, is the one that's used. So dad's copy is silenced, and it's silenced through this antisense transcript. So this is a special piece of genetic material that attaches to the paternal copy of UBE3A and keeps it from being expressed. So the only way that we get UBE3A protein in our brain tissue is from mom's copy. But in the case of Angelman syndrome, these patients have some sort of a defect or a problem or a genetic mutation or sequence variant that makes their UBE3A gene not work properly. Therefore, they can't make the protein, but they still have this silenced copy because of the imprinting. So the body naturally silences the paternal copy of the UBE3A in the brain tissue or in the central nervous system. Now, there's a number of different ways that patients can have a genetic variant in their UBE3A that's impacting it. There's really 4 that we think about. There's some more complex variants, but we won't get into those today. The by far most common is the deletion type. So that is where you have a missing piece, or sometimes a really large missing piece that actually covers multiple genes besides just UBE3A. And so that happens where you just have a chunk that's gone. And so you can't make the protein because the genetic material just simply isn't there. Again, dad's copy is still here, but it's silenced by that antisense transcript, and you don't have a good copy of mom's to make the protein. This affects approximately 80%, sometimes 70% is another range that we think of, but certainly the most common. And these kids tend to have the more severe presentation, more severe clinical symptoms, may even be picked up a little bit earlier than some of the other kids. Then we have mutations or sequence variants, and I think of these as spelling errors. So when we think about our genetic code, there are a whole list of 4 letters that are arranged in a very particular order that make up the code for our genes and the code for how to make all these different proteins. In the case of a sequence variant, you have a misspelling, so a letter that is wrong, or a few letters that might be wrong. And in some cases, that means that the protein is made, but it doesn't function quite properly. It doesn't fold right or do the right actions in the cell. In other cases, it can't make a protein at all because it stops it too early, and so it makes this really small protein that won't do anything. The other 2 interesting ones, though, that you don't always see, and they're less common, and they're also more rare in Angelman syndrome too, are what we call uniparental disomy and imprinting center defects. So with uniparental disomy, remember I said that it's the maternal copy that we use in brain tissue. In the case of paternal uniparental disomy, you accidentally got 2 copies from dad and no copies from mom, and so they both get silenced by the cell. But presumably both of these could be healthy copies that are sitting there. It's just the natural body's mechanism to silence the dad's copy and only use mom's. In the case of imprinting center defects, it's kind of similar but a little bit different. So you do get mom's copy, but in that space where the gene is told to be silenced by the body, you accidentally have a marker that says, hey, this mom's copy might actually be dad's copy. We should silence it. And so the body makes an error and actually silences mom's copy in addition to dad's copy. So altogether, this is a really complex genetic disorder. And kids typically present with neurodevelopmental syndromes and symptoms. So developmental delay, seizures, problems with sleep, or challenging behaviors. But you can imagine a world in which, you've got a healthy copy sitting there. And could we exploit that? Could we use that to our advantage and consider how this might impact different genetic medicines? So it's a really great case for antisense oligonucleotides. So when we have the mom's copy is healthy, we make the protein, dad's copy is silenced. But then you have patients with Angelman syndrome who have a genetic variant impacting their ability to make UBE3 protein from mom's copy, and the dad's copy is silenced from that antisense transcript. But you could have an antisense oligonucleotide that came in and unsilenced the silencer, so stopped the stop. You can block this antisense transcript and allow dad's copy to be used so that these patients can make a healthy protein. And we can also consider things like gene transfer therapy. Now, when we think about gene transfer therapy, you're always thinking in both of these really, is what body tissue do we need to get it to? How are you going to get it to that body tissue? How much expression is enough? And are there any risks of overexpression? Now, you could think about, risks of overexpression when you do know you've got those healthy copies in the background. And could you potentially overshoot and have too much UBE3A? And we do know that there are some syndromes like duplication 15q, which actually have an extra copy of similar overlapping regions as the Angelman syndrome patients. So it's a tight balance. And I think about these things like very simply. You want to think about Goldilocks. You remember the Goldilocks and the 3 bears. You don't want too much, you don't want too little, you want everything to be just right. And so it's really important as we're thinking about all these different genetic mutations, genetic variants, that we're really thinking through all of the steps to make sure that we're confident that we're not going to cause any harm with a potential new therapy. But how exciting is it that we have these opportunities and this technology available that we can start to work on problems like Angelman syndrome?